Good afternoon, and thanks so much for joining us today. Welcome to the 16th edition in our webinar series on the convergence of technology and wetland science. My name is Mike Connors, and I'm the Director of Customer Success at Ecobot. Today's topic is sea kelp forest monitoring and remote sensing. We've got a fantastic panel of experts today with us who will be introduced in just a few minutes. Once we kick things off, we're eager to have a two-way dialogue during the presentations. So I want to direct everyone in the audience's attention down to the bottom of your Zoom window. You should see a Q&A button. Please use that to ask any questions that you'd like to have answered by the panel, and we will triage and field them directly. There's also a chat button, which is more just for kind of one-off comments and things that you don't need to have a follow-up on. Also, just want to make a note that we're recording the webinar, so anyone who registered will get a follow-up email later this week linking you to where you can see the video to that, and also prior webinars that we've done. So our host today is my colleague with nearly 2 decades of experience as a wetland scientist and also the co-founder and chief scientific officer at Ecobot, Jeremy Schewe. So Jeremy, I will hand it off to you to get it started.
Great, thanks, Mike. And welcome everyone. Good afternoon, good morning, good evening, whatever it is, wherever you are. Thanks for coming to join us here again for one of our webinars that we have been promoting since the beginning of the pandemic. It's been a really wonderful journey in drawing together some of the world's leading scientists, technicians, biologists, and specialists in specific taxa. And I'm really excited for our guest speakers here today. They've got a really amazing, fascinating project that they've been working on. And so I want to just kind of cue things up here a little bit before we dive into the heart of our kelp forest monitoring. So since we're looking at a project that is based in Ireland, I had to start off with a writer from the Celtic Twilight, Ella Young, and one of my favorite authors and muses, storytellers, and poets of all time, friends with William B. Yeats. And she had such a deep love for nature and for that which opens us to awe and reverence. And so I feel like this is a great, gorgeous, gorgeous way to start off here. Respecting that which we come from. So this is what we're going to be talking about today. We're going to do a very high-level brief introduction of our presenters, our panelists here in a moment. We're going to take a look at kind of a high-level look at natural and nature-based features for coastal resilience as promoted by the Army Corps of Engineers, both for the United States as well as internationally. Then we're going to jump into the heart of Kate and her team's kelp forest monitoring and remote sensing project, and take a brief look at the wetland data ecosystem and Ecobot. And then we're going to take a look at our next webinar topic. And we should have plenty of time left in the end for questions, discussion. So again, if you just came into the webinar, you have the QA at the button at the bottom. Please use that for submitting questions that might be technical, scientific, taxa related, regulatory related, and we will direct those as appropriate at the end of our webinar. So in a brief share here, this is Kate and her team. So Kate and I have been in communication now. I think it's been about 3 months. And she has been also instrumental in turning me on to some of the wonder and magic of the kelp forests, especially along the west coast of Ireland. So I got to spend some time doing some diving and observation in some gorgeous kelp forests this past September. So Kate is joined by her colleagues, Erin and Stacy, yeah, they've got a great story to tell us and get us ramped up for this really important space that we're talking about, this marine space, the edge of where wetland estuaries and headlands are meeting the sea and what's happening in these places. So we'll go ahead and kick off with a little bit of little information specific to the Army Corps of Engineers recent publications around natural and nature-based features for coastal resilience, of course, with climate change and monitoring of climate change, coastal areas, and the resilience of our coastlines becomes even more and more critical, as well as important for us to be not only monitoring, as Kate and her team are going to be talking about today, but how can we embolden, enhance, restore, and potentially bring some of these elements into the forefront of our understanding, so that we're not so that we're reducing impacts of storm events. So just again, very high level, looking at things from what some of the initiatives that the Army Corps of Engineers is working with, the promotion of their engineering with nature directive, and they're really looking, I'm not really sure what the trademarking is about, but essentially what we're looking at here is an initiative that's going to help develop a more sustainable system that looks at all of the pieces of the puzzle, from the anthropogenic, the economic, the ecological, how do those all dovetail together so that we can make more resilient plans for the future. Also, I want to take a look briefly at some of the recent international guidelines that came out in respect to NMBF in respect to flood risk management. So looking at how coastline bays, estuaries, and freshwater systems, how they all intertie and what we can take a deeper step with there. So a little contrary to most of my normal slides, we've got a lot of words here, but essentially what I just want to bring everyone's attention to is that what we're looking at here in this wetland fringe space, in this marine space, we're looking at areas that over time, due to climate change, have either been underwater, above water, and based on some of the projections of what we're looking at for coastline changes in our lifetime, there's some vital, crucial elements that we need to be looking at as scientists, as policymakers, and then even from the side of planning and engineering, like how does this all tie in, and how can we broaden our ability to make more intelligent planning for the future. And so again, as usual, all of these slides as well as video will be available and recorded for later. And there will be some links in the presentation here for more of this information for those of you who are interested specifically in the effects and what we're looking at here in the United States. But one of the most recent releases came out September 14th from the Army Corps of Engineers, and they have a 20-chapter publication now that can be downloaded online. I'm not sure if you can see my cursor or not, but here in the lower right corner, you've got a website that you can go to get to those publications. And the publication in those 20 chapters is broken down into 3 main sections. So overarching themes, what in respect to the marine coastal interchange, and then taking a really deep dive into specific coastal features, coastal habitats, coastal communities, and then doing the same thing with fluvial as well. And so I just wanted to call out some of those specifically here in terms of looking at this from the wetland fringe, looking at some of the communities that we may be taking a deeper dive into in the next year in respect to some of these webinars, looking at similar, like what we're looking at today, the submerged aquatic vegetation or kelp forests, looking at barrier reefs, islands, coastal wetlands, tidal flats, beaches, dunes, and more. A lot of the headwaters and cloud forest ecosystems are also super vital for protecting our water resources for future generations and for resiliency against climate change. And so my last key up here before I turn it up over to Kate, I want to just tie it back in specifically to kelp forests, and looking at kelp forests in that place of being the ecotone between the deeper open sea and our coastlines, and our estuaries, and how crucial this community is from a nutrient perspective, from a creating resilience for against climate change, which I think Kate and her team are going to talk about a little more specifically as well. But also, I think one of the most important things here is that kelp is a keystone species. It's a critical species in providing habitat, nutrients, hunting grounds for so many species that are either on critical lists or watch lists, threatened, endangered, and even species that are very common. It's a keystone species in the ecosystem. And I think also Kate and her team are going to lean in a little bit to the importance that kelp plays as a natural engineer in buffering against storm events and protecting coastlines and rolling with the effects of climate change. So Kate, I'm going to go ahead and turn it over to you.
Thanks, Jeremy. Let me just, here you go. Okay, so thanks for the introduction. I'm going to be talking about Irish kelp forests in this this meeting. Thank you very much for putting it together. And along with me, we have two researchers that are part of our Kelp Res grant in Ireland, Erin Golden and Stacey Kruger-Hadfield, and I'll introduce them more in their specific portions of the talk. And so what we study is the diversity and resilience of kelp ecosystems. And we're defining resilience in this case as the ability of an ecosystem to recover and maintain function, whether that be structuring the ecosystem as a food source or everything. And so I think that that's a very sexy word right now, and a lot of people are kind of throwing it around. And I just wanted to say that that's exactly what we're trying to indicate in this talk, in our studies. So kelp forests are submerged marine algae. They are structured by submerged marine algae, generally within the family Laminariales, although we include other orders of brown seaweeds sometimes when we're talking about kelp forest forming species. They're found on all continents except for Antarctica, which is just a bit too cold for this kind of seaweed. And Jeremy had some lovely photos of some very common species from the Pacific Ocean. But what we'll be talking about are species within the Laminariales. These are dominant all over Europe, from the coast of Portugal up to Svalbard and Greenland. So they're very lovely. They don't form this kind of canopy that you can see on the surface of the ocean. They form a what we would call in California a subcanopy, but it's a subsurface canopy that forms this lovely kind of chaparral community, if we want to compare it to a terrestrial ecosystem. So fairly short, about 3 meters tall, and very dense, dominating rocky shorelines with moderate exposure. And they're very important ecosystem engineers, meaning they form a house as well as a food source for a lot of species in the marine environment. And they can be very sensitive to a lot of abiotic and biotic stressors. So this is some examples here for you. We have a description of past and future projections for summer isotherms. So this is the warmest the water will get in the kelp habitat, and that's projected to increase, or these summer isotherms are projected to move northward with climate change specifically in Europe, but other regions of the world, which will probably shift distribution of cold water species towards the poles. Biological stressors include overgrazing by herbivores. So this is a very bad picture that I drew from the internet of an urchin barren in Norway. So this is the purple urchin, Sterichinus. I can't remember the species name. It's a purple urchin that kind of decimates kelp forests in its northern reaches of Norway. And they kind of come in pulses. So kelp forests kind of can recover from these events as the urchins die off, for instance, with a viral infection, but they kind of come right back and can decimate that kelp forest again. And it can be devastating for local fisheries as well as just, coastal protection and so on. There's other stressors that includes harvesting, wild harvest by companies or by local users of kelps, increased storminess is another abiotic stressor that's really threatening these communities. So they have a lot of things that are kind of retracting their range worldwide, or moving their range poleward, or just shifting the distribution of kelps in general. So again, this is our study species. It's a laminaria in the genus Laminaria. Specifically, it's Laminaria hyperbrea. In Ireland, it's called cuvee or mayweed. In other regions, like in the UK, it can be called tangle, but that can be confused with Laminaria digitata or oarweed. It's generally in the subtidal, and it can live about 10 to 15 years. So fairly long-lived, just as long or longer than the giant kelp that you find in California. And what's interesting and unique about this species, which I think is really cool, is that you can age it quite like you can age a tree. So it has these kind of tree rings that are created by prolonged winter stages. So their growth is reduced. You get a darker band, lower nutrient availability and such will cause these kind of bandings. So the oldest individual we found in Ireland is around 15 years old. In Norway, they can get to 18 years. So they have the potential to stay and structure ecosystems for a very long duration. And like all brown algae within the Laminariales, they have a very complex life cycle, which we are going to touch on when we talk about resilience and recovery of communities. So they have a sporophyte stage, which is what we consider the kelp. This is what structures ecosystems. It's the macroscopic kind of form of this species. This is diploid, meaning it has two pairs of chromosomes, quite like us. It forms these reproductive structures on its blade, where meiosis occurs and produces these haploids or single chromosome set spores. These are mobile, but they're microscopic, so they're not going too far unless there's really strong currents. They're about in the water column for about 24 hours, and they settle out in the benthic environment, so bedrock, stones, what have you, and develop into gametophytes if they're lucky. These are basically the sexually, I won't say sexual stages, but you can have male and female gametophytes that produce gametes that recombine in sexual recombination. So quite like ovaries and testes, they're producing an egg and a sperm that then recombine to form this sporophyte. So that would be recruitment. A fertilization event would allow recruitment of the sporophyte, and then you'll get this macroscopic forest theoretically the following year. So fairly complicated life cycle with two crucial stages, the macroscopic and the microscopic stage. And this is a seasonal pattern, or these different events within the life cycle happen on a seasonal scale. So for Laminaria hyperbrea, which is different from all the other kelps in this habitat, zoospore formation will occur in the winter, gametophyte development winter to spring, and the new sporophyte generation generally starts appearing spring through summer. And that's very important for the latter part of this talk. Other species will have this process start occurring summer through autumn. So if you're going to do an experiment in the system, it's quite handy in terms of identifying what's recruiting, developing in your ecosystem, because something unique to the Laminariales, or maybe not, is that they all look alike when they're really little. Frustrating if you're trying to do benthic ecology in situ. So just lastly, what I'll touch on is that this species has experienced past range restrictions. In the last glacial maximum, it's projected to have retracted its range all the way down to the southern Irish coast, the southern UK, Brittany, and Portugal, down to Morocco. Modern day distribution is from the northern portion of the Portuguese coastline all the way up to Svalbard, and this is projected to retract northward all the way to the mid-UK southern Irish coastline in the most extreme RCP projections from the IPCC. So theoretically, climate change is very threatening to this cold water kelp species. So in our research, we're really interested in historically where kelp has been found in Ireland, specifically just Ireland, because we're trying to manage this resource in a national scale. And, what has hyperbrea been used for? how have people recorded it in the past? How are they interacting with this species? And we're very interested in two aspects of resilience with this kelp. So one is genetic diversity within the populations. What is the genetic structure and connectivity across populations within Ireland? And the other is there a natural resilience within this life cycle? Remember, from macroscopic to microscopic stages. So do they form banks of microalgal stages, quite like seed banks in terrestrial environments, that allow these communities to recover after a major disturbance? And then lastly, we really are kind of trying to develop monitoring protocols, monitoring systems, tools that people can use, maybe people who aren't specialized researchers, to kind of contribute to data sets on kelp forest presence, potentially ecosystem health, and just distribution shifts over the next couple decades. So Erin's going to really talk about that as we move on. Again, that kind of just feeds right back into how we have a historical, or, where Hyperborea systems can be found in Ireland. We use citizen science data sets to create our kind of assessment of historical distribution. And I think that any future monitoring is just going to be incredibly valuable to future research programs. So I'll just kind of briefly touch on historical records. So these are very hard to find, which is not crazy, right? Kelp are very large, and they're very hard to preserve unless you have them in formaldehyde, and then they're not exactly useful from necessarily a genetic perspective. They can deteriorate, lose their color, so they're not that useful from a morphological perspective, like for identification purposes. And they don't dry out well on herbarium paper. They just mold because they're so big. So we can't really find a lot of information about kelp in herbaria. There's not a lot of information from old textbooks. So we went to the Linnean Society in London and kind of scoured older records. The UK has a really nice history of marine biology, but a lot of it would be intertidal. So these are subtidal species. We don't have a lot of information unless things were pulled up on anchors from the Naval Academy. People have some records of that, but you can't really tell about established ecosystems with this kind of data. So the earliest record we have of Hyperborea is actually down in Wexford County here on Hookhead Peninsula. So if you guys have ever been to Ireland, this is a very lovely lighthouse, very unique geological feature on the southeast coast. And people have repeatedly sampled this region, but it took quite a while for people to kind of proliferate their sampling across the country. So if we look at the next kind of decade of recording, we don't really get anybody kind of talking about kelp until the 50s and 60s again. So that's the light pink dots around the coastline. There's very few of them. We really get a big pulse of records in the 1990s when there was a research project called Biomar, whose goal was to record subtidal habitats in Northern Ireland and down through Ireland, if you will. And then the proliferation of a citizen science group called Sea Search Ireland, which is quite like Sea Search in the UK or Sea Search South Africa, has really kind of expanded our recording of kelp. So that's from 2000 to present day. And it's just like a big boom of records from, it's kind of like the red-orange thing from Northern Ireland all across the country. And so really, I think the development of technology like scuba diving, the development of things like the internet where people can share these records online has really helped us better understand distribution. But there's a few caveats there. So sites are rarely repeatedly recorded. It's like once somebody finds a nice kelp forest to dive in or to swim past as they're going towards a deep reef or a wreck, something like that for their recreational dive, they don't really record it again. So there's no information on ecosystem health or whether the ecosystem is still present. And we also can't tell if these records are living kelp. So there's no indication of kelp health, whether it was fixed to the bottom, whether it was stranded on the beach line. Was it a kelp forest, or was this a random record that, just happened to recruit to that site? There's not a lot of data on ecosystem health, abundance, or anything like that. So that's something we're really trying to move forward with when it comes to citizen science records like scuba divers. And the other thing I kind of want to mention is that, we do have some data about kelp from a cultural perspective. It's always been very important for agriculture, food, local economies, especially in Connemara and different regions of Gaeltacht, where kelp was used to create potash as a fertilizer and so on. And then we have some cultural records. This is an herbaria record. I think this is probably oarweed, not actually laminary hyperborea, that was created and made into a rosary. So, it is very important in Ireland. It's just something that we kind of need to be able to record in a better way. And so the last thing I'll say is that there are some hotspots for recording, and these generally are centered around institutes of higher education. So we have Galway, we have Cork, where you have major universities. Not a lot in Dublin, where you have major universities, but that's a very high sediment area where you just don't find a lot of kelp. So we do have this kind of bias in our data sets that maybe we need to expand across the coastline just through general outreach activities. So I'm going to just touch on monitoring and how we do that, and why it's kind of easier to try to segue from why it's easier to do with a higher education institute than it is with citizen science kind of groups, because it is it's a very detailed, in-depth, time-intensive effort. So what we have been aiming to do over the past 5 years is kind of create a modern baseline for kelp forest ecology in Ireland, create monitoring tools, and we've been contributing all of our data to international networks, and then try and develop long-term data sets that understand, or define the resilience of these habitats in Ireland. So what this required, and unfortunately we haven't been able to maintain it, is establishment of monitoring locations with subsurface buoys that wouldn't get picked up by fishermen. We put data loggers out so we can record environmental conditions that are found within kelp forests, and that might help feed into modeling efforts if we were to go that way. We have recruitment habitats to see what preferentially settles into these ecosystems. So, you probably know a little bit about different types of animals have different life strategies within their life cycles. Some are pelagic, some are benthic recruiters, trying to capture what is selectively settling into kelp forests or what is bred within kelp forests using these kind of recruitment habitats. We look at succession processes, so how kelp forests recover, and that kind of feeds into some later slides. We do biodiversity transects, just basically counting everything that we find in a certain swath of area in these kelp forests, and that helps us define, what is living in these kelp forests seasonally, annually, across different depths, across multiple locations. And then we look at the kelp itself, trying to define productivity. And what I'll show you really today is just biomass and age of kelp forests, but you can do a lot of different things. We've looked at pigments, we've looked at phycocolloids, we've looked at proteins within these communities that will then feed into food webs in the nearshore ecosystems. And then we look at growth rate of these individuals to try to determine, how is this biomass produced and what's the rate of production across these systems. So what I'll tell you really quickly is just we've looked at kelp forests, but we also look at other nearshore ecosystems to get an understanding of how important kelp forests really are. So compare them to saperlid worm reefs, which form these balmies, quite like coral reefs, marl beds, and just sediment bottom habitats where scallops and rays are quite prevalent. And then rocky, kind of rocky bottoms and such. So areas where you have a lot of sponges and other filter feeders. And what we see is that kelp forests are quite unique from all those other habitats. So they really stand out. The biological community that's associated with them is very unique. The other habitats cluster quite well together as to themselves, as well. So each habitat is kind of defined by the biological community that's associated with it. And that's quite nice. But even within kelp forests, what we see is that different sites kind of cluster out with themselves. So each site is unique. Each kelp forest is actually unique within that type of ecosystem. So you have a lot of regional differences that impact which kelps are, or what the communities are going to look like. And that may be an aspect of just the local environment. That may be what is available in the recruitment cohort. So what is current systems are delivering to the local site. But it's very important if you're thinking about monitoring and developing monitoring plans for a coastline or the country. So quickly, I'll just talk about what species really define kelp forests, what may be called indicator species in healthy kelp ecosystems. So you have the blue-rayed limpet, which is very distinctive, selectively recruits 2 kelp species, so oarweed and cuvee, Limera digitata and Limera hyperborea. It's generally found at the base of the blade from spring to summer. You have a lot of echinoderms, so you have a lot of sea stars, sea cucumbers, you have sea urchins, but never at a level that might cause destructive grazing. So we don't really have the purple urchin that you see in Norway. This is what we call the common urchin. It can be quite big and white. And we have a lot of gastropods. There's also a lot of filter feeders and tridivores in these communities, suggesting that they're kind of taking up kelp organic carbon, which is cool. And then seasonally, we see a lot of really important commercial species. And now they're not in high abundance. You don't get like a mass of crabs in one area. But we do see them kind of coming into the habitat and utilizing kelp as kind of a pedestal to feed in the water column or as shelter. For instance, lobsters quite often are found in the nooks and crannies of rocks. And fishermen will selectively place their crab or lobster pots inside kelp forests to get their daily catch. We also see in the summer a very large pulse of juvenile gadoids, and gadoids are commercial fish like cod or pollock, and these yellow dots are kind of just covering or circling these little fish, and they're very, very tiny, very hard to identify as species, but super abundant just above the kelp canopy here. And you notice you can't see them, and maybe that's a very nice camouflage from predation for these species. Now, I'm just quickly going to touch on productivity. It's a very, very messy graph, but basically we get the highest kelp density, which corresponds to highest biomass. This is interesting not only from a food web perspective, but from potentially applied uses of these kelp ecosystems. I mentioned before, people are very interested in harvesting kelp, and they do this in France and Norway. And this is just really starting to become a commercial industry in Ireland, which is very contentious. So you see, highest densities kelps in shallower depths, which are also the stonier regions, 2 to 4 meters, lowest astronomical tide. This is also where you see kind of your older individuals. So you're getting these older communities, maybe meaning that they're more stable at these depths, developing around these kind of optimal depths for harvesting. So it's kind of an interesting trade-off. And this is just a nice kind of show that this is kelp forests can be quite like terrestrial forests, in that once you get really old, the density of individuals really goes down because you have this canopy shading effect. And so once these canopies are cleared, you get these high kelp densities again, where younger individuals grow up and then they happen to self-thin over time. So we're really just kind of describing the community in that sense. So now I'm going to jump to Stacy, she's a molecular ecologist at the University of Alabama, Birmingham. And she's been working with us in Ireland for 4 years now, I believe, and trying to define genetic diversity and structure in these communities.
Thanks, Kate. In my lab at UAB, we use a variety of tools, and for the KelpRes project, those tools are what are called microsatellites, and they're repetitive regions across the genome that, until fancier technology came along, were used in paternity analyses and in CSI type shows to figure out, who the father is or who a criminal might be. And they're really polymorphic and variable regions across the genome that we can use as a tool. And they're similar to what are used for 23andMe or Ancestry.com. If you've tried to figure out what your ancestry is, we can use those same principles to figure out the structure and connectivity of any type of organism. And so on the map that Kate has on the slide, there's little green dots that show the different types of sampling that we've done, and what is emerging from the population genetic literature is the importance of temporal sampling. So Kate mentioned earlier that a lot of the herbarium records, there's sort of a one and done sampling approach. And this is what has been done for population genetic studies as well. And it's really problematic if you want to understand the resilience of populations, understand how genetic diversity is changing. And this is particularly relevant if there's going to be any sort of wild harvest. There's a lot of talk about the sustainability of seaweed harvesting and of seaweed aquaculture, and we really lack a lot of information that we possess in agricultural contexts of corn and other types of plants that we've been cultivating for thousands of years. We just simply don't have that information for macroalgae. And so in the plot in the middle, what you can see is geographic distance on the horizontal axis. And then on the vertical axis is genetic differentiation. And this is just a measure of how similar or how different populations are. So when FST, genetic differentiation, is 0, it means you have the same frequency of what are called alleles. So different variants across a genome and at the particular loci that we're looking at. And as FST increases to 1, you're becoming completely distinct. So you have a totally different set of alleles at a given locus. And this is sort of in contrast to what is typically discussed in the ocean, that the ocean is this big mixing bowl. It's this big open environment where everything is everywhere and there's lots of gene flow. And what we see in macroalgae is this is rarely the case, particularly in species that can't raft. So if you're familiar with Macrocystis, the giant kelp off of California, it has these air bladders. It can raft great distances. That's not the case in something like Laminaria hyperbrea. And so what we see is a really strong relationship between genetic differentiation and geographic distance. So as you move further from one population, you become more genetically distinct. And this makes particular sense along the Irish coastline, which is incredibly rugose and has a lot of structure. So Kate, if you want to move to advance to the next thing. And then the plots that you see down on the bottom are a different type of analysis. So it's called a discriminant analysis of principal components. You don't need to get bogged down in the math behind it, but basically any points that are close to one another are genetically very similar. Any points that are further away are genetically more distinct. So it's just another way of looking at genetic distinctness. And these are analyses that don't make any assumptions about what's happening in these populations. And so they're pretty powerful tools to understand more about genetic diversity and genetic structure. And you can try to figure out how many what we call clusters, so these different colors that you see on these plots, how many there are. And we see pretty strong statistical evidence for these different colors that you can see drawn on the map. So the red down in the southwest, the blue on the west coast of Ireland, the more yellow in the northwest, the gold on the northeast, Eastern Ireland, and then the southeast. And so you read these plots by first looking along the horizontal axis. And so we can see that there's pretty strong separation between the southwest, the southeast, and the east, if you're looking in that first panel, and from the west, the northwest, and the northeast. And then you read the plots by looking on the vertical axis. And then we see separation between the south and the east. And each of the different axes are just different, what are called principal components. And so it's just a different way of looking at this multidimensional space to understand more about genetic structure. And these data show that, like many other macroalgae that have been studied, there's a lot of diversity, but it's very highly structured based on where you are. And this would have pretty important management implications. So if you, next slide, it's like we're going back to a slide deck. And one of the tricky things about using these genetic data is being able to compare to other data that other labs have generated. And there is some subjectivity to what you call an allele. And so we worked with actually the lab where I did my PhD with Miriam Valero's group and with Esther Serrao's group in Portugal. to calibrate our data so that we could compare to the existing repository of genetic data across Europe, which is pretty important to contrast different areas of a distribution. And so what you can see in the panel that's in sort of the upper right of your screen is a different way of looking at a discriminant analysis of principal components. So this was from a paper that we published last year in the European Journal of Phycology. And we only had, I think, 5 or 6 or 8 populations, I can't remember, that were in County Galway, Donegal, Clare, and Cork. And so we can see strong structure in Ireland, but what you see across that first principal components axis, so that first horizontal axis, is that Ireland is separated from the previous data that had been generated along the Brittany coastline. So basically from Mont Saint-Michel down to southern Brittany near the Quiberon Peninsula. And what we see are all those points in France are clustered right on top of one another, which means that they are genetically much more similar to that spread of points that you see in Ireland. And this is curious because the Brittany coastline is pretty rugose as well, but we see different structure. There's a lot more information known about Brittany, and there's a pretty strong kelp harvesting operations in Brittany. And so understanding where there's centers of diversity, understanding the life cycle, which I think is what Kate's going to touch on the next slide, is really critical to be able to make informed decision making at a management perspective, at a conservation perspective, and then translating really complicated math and complicated statistics to the people who make policy. And that's one of the, I think, the challenging things, but without these data, you know, that's not possible. So I'll give it back to you, Kate.
That's great. So yeah, I am going to start talking now about, I mentioned this before when I was talking about the productivity and the biodiversity of kelp ecosystems, but succession. So how communities recover from, for instance, a major storm disturbance that wipes out the whole sporophyte canopy, or something like harvesting. And so we did this small experiment looking at native succession plots, which just means that we cleared a plot that was just bedrock, coral, and algae, which kind of, it looks like a pink rock if you've been to the Intertidal. And you know what, we scraped all the inverts and all of the visible seaweed off the top of this bedrock area and monitored recovery. We also put out a similar area of concrete slab. So this is our sterile succession plot, and kind of we conditioned it in seawater before we put it out, just to leach all the cement, you know, the chemicals out of the cement that had been, you know, that are just embedded from manufacturing. And then we plopped it in the water, and then we looked at both communities as they recovered over time. And so I'll just show you what it looked like after 100 days after clearing. You get a lot of juvenile kelps on native plots. After 208 days, these start to self-thin. The animation didn't work, sorry. They undergo self-thinning as these kelps get much, much larger. On these sterile plots, after about 40 days, you get maybe a nice biofilm, turf community sort of building up on the plot. Turf meaning just microscopic or filamentous algae that form this kind of film on the top that captures sediment and sand. So you have this kind of, not a biofilm, but you have something starting to grow, not kelp by any means. Then we start getting invertebrate recruitment. So again, this is just calcified worms. You start seeing tunicates and sponges settling on it. And then after a full year, we start getting kelps. And so what that's kind of indicating is that after a full year, you've had a full, you've had the opportunity for kelps to produce these spores from the sporophyte, let those spores settle, produce gametophytes, and then these, you know, recombine and produce juvenile sporophytes, which we're starting to see. This kind of indicates to us that there is this microscopic bank of kelp stages already present on bedrock that we can't see, that we couldn't clear. And that kind of leads us to our KelpRes project, where we wanted to really look at how that bank factored into kelp ecosystem resilience. So we took a 3-pronged approach to really target this. It's very tricky because you can't bring you can bring a microscope underwater, but we were not about to develop one of our own. There's instructions for making one from Mike Nuschel. He did a lot of work in California. from the 60s to the 80s, was actually my PhD advisor's PhD advisor. And so it's been developed. We just weren't going to make it again. So it's kind of tricky to find these banks. And the way we were trying to do it is we put out settlement tiles, and these were cement blocks that held microscope slide racks. And we left some of them in situ for a full year, and some of them we took out seasonally to capture seasonal reproduction. Again, these kelps reproduce at different times of the year depending on the species. So Hyperborea in the winter, all the other species summer through autumn. We also took bedrock out of kelp forests, some monitoring sites, and we tried to grow kelps in an aquarium system that was mimicking environmental conditions. So we thought if we remove the canopy, just like a storm or harvesting would, these microscopic banks would just grow up out of there really quick in these artificial environments. And then lastly, we tried to barcode and then meta-barcode both bedrock samples and these microscope slides from seasonal and annual experiments to see what we find in the microscopic boundary layer. We also took these microscope slides and we looked at them underneath a microscope to try and count gametophytes, zoospores, and everything that was settling seasonally and annually. And now, again, I think I mentioned this when I was going over the life cycle, but you can't differentiate the species of kelp based on these microscopic stages. They actually look identical within the order. So that's where the barcoding comes up. We have microscopy data telling us abundance of either zoospores or gametophytes of certain sizes. And the barcoding can feed into what species those might be. So I just have a small video just to show you what these kind of look like underwater. Hoping you guys can see. And we have annual slides here on the right, so you can see they're covered in a nice biofilm that also has sand on it. There are a lot of kelps actually growing on those annual slides, so we know that the experiment worked. It captured reproductive events across a year. And then we have our seasonal slides. These are getting collected at the end of August 2020. So these would have captured any seasonal reproductive event in summer. So we would expect these to have, for instance, oarweed or sugar kelp on them, but the barcoding could actually feed into that information for us. So what we found is that if you look at the density of different stages, so our zoospores are purple. We have 2 cell size gametophytes, 3 cell and 4 cell. They actually don't develop larger on these microscope slides. We didn't find any that were larger. And you find different abundances depending on the depth of your experiment. So shallower is closer to your conspecific or congeneric species, Laminar digitata, Saccharina latissima, the sugar kelp. You're getting a very large abundance of zoospores, and then more abundance of gametophytes in those areas where you have more species present. And then lower abundances when it's just the laminary Hyperborea canopy. So that's kind of interesting. And we see some seasonal bursts in different, in zoospore and gametophyte densities as well. So if we look at winter, you have a lot of zoospores, presumably from Hyperborea. Summer, spring, you have a lot also presumably from Hyperborea. You have more gametophytes than zoospores. And then autumn, you have this kind of burst, and that's likely because you have multiple species kind of contributing to this community. If you look at annual slides, you probably have all those species captured there, a lot of zoospores and a lot of gametophytes. So hopefully that's our bank. And if we look at the barcoding data, so it's presented as, seasonal as well as across 2 different sites and 2 different depths and canopy levels, all the different factors. If we look just at these kind of MKUPLH, what we're finding is that you get a lot of laminary digitata, for instance, in autumn. In winter, you only get laminary Hyperborea. In summer and spring, you get a large mix of different kelps, some of them that we don't see, we don't ever see the sporophyte of. So for instance, you have Alaria esculenta at this one site. This is a very This kelp is not really found in County Galway, where the site is located. It's further north or further south, really likes high exposure, but it's gametophyte or potentially just a zoospore was present in the spring at the site. Just never see it. We also have an invasive species, Undaria pinnatophyta, which is generally only found in harbors and such in the UK and now Ireland. So we never see the sporophyte here, but it's interesting that a microscopic stage could be found. We also found that there's a lot of mixed kelp, so we get very messy sequences back from this barcoding effort, and that might mean, so we have to kind of still work through the metabarcoding data, and that's going to hopefully tell us a lot more about the diversity of species in these microscopic banks of kelp stages. So I'm just going to touch on our citizen science efforts. We have these dive slates for people to kind of go out and record ecosystem health that we've delivered to local Irish Underwater Council groups. So these are dive clubs. They don't work quite like dive stores in the US. They're local groups. Everything is volunteer based. And just people who like to dive together in a certain region. We have the Galway Sub-Aqua Club, and we have a university club in Galway called the NUIG SAC, so just NUIG Sub-Aqua Club. And hopefully this will start getting utilized more, and people will contribute these records to our online submission platform, so we can keep continually recording sites that people use for their recreational dives over time. And that can feed into a very robust historical data set. But now I'm going to switch over to Aaron Golden. He's a professor at the University Galway or NUI Galway, and in math, statistics, astrobiology, genetics, all things, jack of all trades, master of many.
Okay, nice ending there. Appreciate that one. Yeah, okay, so I'm going to talk briefly about, I suppose, the other end of the scale, I suppose, to go from the kind of the micro to the macro. One of the important issues that we were asked to address in this particular project that was funded by the Irish Environmental Protection Agency was to try to get a sense of how tractable it would be to, in a cost-effective manner, monitor what we've been studying. I mean, not just monitor the specific sites that Kate and her colleagues have been diving in, but monitoring around the entire crinkly coastline. And that's a non-trivial problem. My colleagues have been using the big word rugosity recently. It's another way of saying that the coastline is a mess. I mean, it's very, very windswept, storms regularly roll in, it's extraordinarily rocky. It's all the beautiful stuff that the Irish that the Irish tourist industry thrives on, and rightly so, but it poses a very challenging problem for us. There's no question of diving everywhere. There's no question of flying drones everywhere. Is there a cost-effective way for us to actually map and monitor? And I suppose this opened up the possibility for us to explore the availability of Earth observation data. Of which there is a substantial amount. You're all aware that Landsat 9 went up quite recently. And that's, we're talking about almost 30 years worth of data that have been collected by the US Geological Survey and NASA, and that are freely available to all. The problem with the Landsat program from an Irish perspective is you may have noticed that I have the pallor of a typical Irishman. There's a reason for that. We don't get the sun a lot here. The weather's usually terrible. I mean, it's still a beautiful spot, but the weather's usually terrible. That's a problem because when the satellites are passing over, 9 times out of 10, it's cloudy. There's that problem. The second problem is the resolution. The spatial resolution that we're talking about in an Irish context is not the sort of spatial resolution you get off the coast of California or the coast of South Africa or Australia, where you get these vast canopies floating in the ocean, which basically means that you'll be able to pick them up no problem, and into the bargain, because they're floating on the surface, you can apply the same kind of tricks that folks use to characterize land vegetation to monitor, study, and so on and so forth. The problem we face in an Irish context is that the kelp areas are quite close to the coastline. So there's a sort of a ribbon of a boundary, if you like, of spatial resolution that we're kind of restricted to. And secondly, as my colleagues have pointed out, they don't float on the surface, they're below the surface. So automatically you can't use the tricks of the trade that have been typically used for the California and other kelps to try to monitor them. So the issue there was, well, what could we possibly use? European Space Agency and the European Commission has funded the Sentinel program. They have much higher resolution in terms of spatial resolution, so they're down to 10 meters. And also they pass overhead maybe once or twice a month, maybe 3 times a month. So you have a better chance of getting a good shot. But again, the problem then we faced is that we wouldn't get the right kind of resolution. So fortunately, science, and in particular, a company by the name of Planet Labs has sort of come to our rescue. You may have heard of Planet Labs. They're kind of I suppose it's an overused word, but they're basically a disruptor in the remote sensing space. They've been really taken the Earth observation industry by storm by pioneering the use of CubeSats. So we're talking about spacecraft the size of what are these crisp, or what do you call them in the States? Not crisps, chips, right? These chips, chip containers, right? I forgot my name of them right now, off the top of my head, right? So you have a spacecraft that's basically a glorified telephoto lens with solar panels, and you can launch a couple of hundred of them in one go. And so this company called Planet Labs, they have about 200 or 300 of these things in orbit. So this means that these things are passing overhead about maybe 2 or 3 times a day. And so from an Irish context, not only are you very likely to get a free shot because there's going to be a gap in the clouds, but also you're going to get good resolution. The resolution we can get is 3 meters. So on the right-hand side there, you can see an image of one of the dive sites that Kate and her colleagues dive from. And what you can see there is the imagery that you can get from the actual spacecraft. And it's actually good enough for us to compare, if you like, the transect dives, and I've marked them in the red and the purple line 1 and 2, where Kate and her colleagues would actually go out and do a kind of a survey. We can actually study what they are likely to experience by analyzing the data that you're looking at. So the panel in the middle there, you see their PlanetScope data combined with GSI MI bathymetry. The Irish government's Geological Survey of Ireland and also the Marine Institute have been mapping the entire seabed, I guess, off the Irish coast all the way out into the deep Atlantic. And that's been absolutely invaluable for us, because what it's allowed me to do is it's allowed me to take the data from the spacecraft and to overlay the bathymetry. And so as a consequence, I can actually emulate a dive that Kate would actually take. So what you can see there on the right hand and in the little subplots that you see there is that what you're seeing is bathymetry, and also what you're seeing is a ratio in 2 color bands, which are a proxy that we use to bring up the reflectance of the actual kelp structure itself. So when you see this RG value up at a certain value at about 0.9, that corresponds to what Kate would be seeing as bona fide kelp as she swims through that transect, whether the transect is 1 parallel to the coast or 2 straight out to the center of Cashla Bay. And the important thing about that is, first of all, it speaks to the fact that we can actually survey the kelp using these CubeSat data sets to the extent that we can put some constraints on where the kelp actually is. To move forward to the next part of the slide there, Kate, the next stage that we can do is we can then apply some very straightforward machine learning algorithms to say, well, if we know the bathymetry and we have a rough idea where things are, if I was to ask you, Mr. AI algorithm, if you could classify structures and patterns, what structures and patterns that you see would we know for a fact correspond to where the kelp actually is? And what you see there are 3 plots which correspond to increasing depths into the water column. And the red dots that you see there correspond to those particular pixels that the AI algorithm responded to as, I think this is the thing that you think is kelp. And they actually correspond extraordinarily well to the actual kelp beds that Kate and her colleagues would routinely dive through. So the first thing we were able to show from a feasibility perspective is, yeah, we can actually do this monitoring thing from orbit using this completely new innovative class of remote sensing platform. The second thing we could do is we could put some limits on how far we could go down to. The water column in Ireland is fabulous if you're in the marine life business, simply because it's so rich and abundant. That's a bad thing for us, obviously, because it means the water column is going to be full of interesting nutrients which block out the light. So in other words, there's a limit to how far we can go down. And so we can make a reasonable estimate of seeing things down to 10 meters. Once you go beyond 10 meters, you start to lose structure and things break up. And that's the difference between the slides, the sub images you see there and the left-hand side in the middle and the one on the right. The one on the right is when you go beyond this 10-meter depth line. Having said that, the new generation of CubeSats that Planet Labs are producing are going to have new spectral bands. So the way in which I like to think about it is these spacecraft take pictures with a number of different sunglasses on, if you like. And what they do is you combine the different images to bring up structure. So if anybody's ever played around with a straightforward camera and has a yellow filter in front of it, or has even worn different types of sunglasses that really bring out contrast in the clouds and so on and so forth, you can use exactly the same straightforward magic, if you will, to bring out the structure and contrast that you can actually see from below the water column. So we're hoping with the next generation of these satellites that Planet Labs are launching that we'll be able to probe down deeper and expand, if you like, the scale at which we can actually survey and monitor the kelp right now. And I guess the really interesting thing is, if I can go off on a little bit of a noodle, is that you can use some very, very cool dark arts in machine learning to, if you like, take the data that we can work with at a high resolution here and roll it back onto data taken with the Landsat spacecraft. So we can use that. I think the really interesting thing here, as I'm getting to, is that we have like 30 years of data or more from the Landsat program. We don't have the resolution, and we have spotty coverage. If we can ground truth everything right now, we can go back in time, and to some extent we can track the predictions of how the kelp is going to respond to the rising temperature in the actual ocean. So that's, if you like, the promise of what we would hope to be able to do. And what I can tell you is that based on the preliminary work they've been doing as part of this project, it waddles and it quacks. It's definitely a duck. And so it's definitely something worth investing in. Thank you.
Awesome. Okay. Well, I'm just quickly going to move on to talk about governance and legislation that pertains to kelp forests in Ireland. Currently, there are not really any regulations relating to kelp ecosystems. There are regulations over fisheries and nearshore ecosystems, but nothing specifically pertaining to kelp. Although the Water Framework Directive and the Marine Strategy Framework Directive do include a lot of text on good environmental or ecosystem status within these habitats, or marine habitats, coastal habitats, and water ecosystems. So moving forward, KelpRes is part of a global report on kelp ecosystem health. I am contributing to that, and hopefully with the Green New Deal in the EU, we're going to start putting these ecosystems kind of in the limelight. I know that they are probably one of the ecosystems that's most in the limelight worldwide after coral reefs and seagrasses, but they're just not quite as protected as they are in regions like the US, where we have a national park, for instance, Channel Islands in California. We have a very nice MPA network along the coast of California that goes all the way up to Washington, that actually helps these communities kind of reseed adjacent communities that are highly fished and so on. We are working with the EPA and National Park Service in Ireland to invest more in monitoring. The EPA has a new actual job for coastal monitoring, subtidal ecosystem habitats. And so that's really cool. And hopefully the tools that Erin's been speaking about in our dive slate will actually be used by recreational divers, school courses, and citizen scientists to kind of create a nice record of everything moving forward. So I'm going to just finish out with this lovely quote from Joni Mitchell. And then I have a nice link here for the Sea Search Ireland group. If you're interested in helping foster subtidal research, they're the only group that's actually doing it, including academic, after KelpRes finishes, because we are supported by a grant from the EPA that has it's ending in November. So any ongoing subtidal research in Ireland is basically just through Sea Search Ireland, and you can fund them here at this GoFundMe link. And that's it. And so if we have time for questions, I'm not sure we will, because I think we've gone 5 minutes over, but we'd love to take some, and I'm sure that Jeremy will supply these slides and our contact information if you need.
For sure. Yeah, great. Thank you, Kate and Stacy and Aaron. I mean, great stuff. Fascinating, Aaron. We're going to have kids doing remote AI diving into kelp forests here in the near future. So Kate, got to make sure that you get some good videos so that we can build the real deal here. So thank you everyone for sticking around with us. If you've got to go and get back to whatever you're doing, please feel free. Please join us again. November 17th is our next webinar, and we will be focused on wetland mitigation, monitoring, and planning tools that are being used here in the United States and abroad. So if anyone does have any question that's going to stick around for a couple minutes, we will take a couple. I have a few that I went ahead and wrote down. If you have anything, throw it in the QA channel there. But one of the questions that I wanted to ask you, Kate, was in respect to, you were speaking to resilience, and you gave a great definition of it in respect to kelp. And so my question is twofold. One is, you're talking about in Ireland some exploration with utilizing kelp as an agricultural product, and so doing more cutting around that and looking at that. So first question is, how long does it take for the kelp to recover if it's cut, or does it recover? Does it just need to be reseeded?
So there's a lot of work. Specifically, France and Norway have had these industries, mechanical harvesting industries, for quite a long time. And in Norway, they do it maybe more intensively than France. So there's been some very nice research showing that even if you leave the region fallow for over 5 years, the ecosystem does not recover. The kelp itself grows back actually very quick. So once you remove canopy, new individuals can grow to the same size as 3-year-old individuals within, their growth cycle. So they get quite big very fast, maybe like a meter tall, but they're by no means mature. I didn't really mention that these kelps also are like a substrate or substrata for hundreds of individual, you have amphipods, small crustaceans, sponges, other seaweeds, tunicates, all these things kind of colonize the kelp itself, the holdfast and the stike. So it's important, not because it's creating a forest, but it's also, things are really living on it. And the 1 to 2 year old individuals really don't have anything growing on them. It's once you get to 4 and 5 up to 15 years old that you're really hosting a very robust, diverse kind of epibiont community. And that just disappears once you start taking these kelp, the canopies out. So it's definitely going to be over 5 years. That's the legislation in Norway recommended, leave these sites fallow for 5 years so that the community can recover. But by no means does it really resemble what it was before harvesting. I think that's probably true for most crop situations. But we haven't been able to do that work in Ireland specifically.
Do you have any numbers that you could compare that to in respect to storm events? Because, just kind of tying into what I spoke to briefly at the beginning of our conversation here today, looking at how communities like this can help with coastal resilience as a community en masse and buffering storms. Like, what are you seeing in terms of recovery from large storm events?
The difference between storm events and large clearances by harvesting is that you're taking out every individual in a community in a certain area. You're taking out all of the large sporophytes that are reseeding the community for the next year. And while there's probably a microscopic bank of these stages that allows the system to recover, or maybe they're harvesting after they produce these zoospores for the next year's, large apparent kelp. When a storm takes communities out, it's very heterogeneous. So you have certain spots within a kelp forest that get wiped out. It's not a clear swath. You have a very large mix of different age groups contributing to the next year's population that's going to regrow and recover after that storm event. So it's just a very different seascape when you're harvesting versus when there's a storm, because you have a lot of local adults helping the community recover in a storm, whereas you have nothing with a harvest. And of course that, you could adapt your harvesting plan to allow for heterogeneity. within the site where you're harvesting, if you want to do that. But we actually tried to kind of create an experiment this year for a master's student from Belgium where we could mimic, two different types of harvesting approaches, storm events, and then just a natural population, and see how that community recovered. However, subtidal ecology is very difficult. And I think that storms or other things kind of took out all of our markers, and we weren't able to kind of refine our plots. We found 2 of them out of 20, which is it's not good enough for science.
So then all you had was storm data.
Yeah, sort of.
And nothing to compare it to. Right.
Yeah. So I think it's an area that people should move forward with and keep, trying to do that kind of work. It's just, I think you need more secure plot markers than what we started out with, just for one. There's probably more experimental design flaws with that.
So one of the things that I've been looking at over the last 17 years now is the migration of more southerly species on a terrestrial or freshwater wetland habitat in the Arctic, mostly up in the Scandinavian countries. I'm interested both from you, Kate, as well as from Stacy, like what are you seeing from that side of things in terms of southern species starting to move north into Ireland and into the North Sea, etc.?
I'll start and Stacy can jump in if she wants. We have seen some warmer water kelps move. We don't know if they're moving. They're not actively going necessarily to a certain spot in Ireland, but we did find what we call the golden kelp, which is more of a warm water Mediterranean species. It's found in Morocco up the Portuguese coastline, and now in the UK. We had the first record of it actually off of Balmullet in Mayo, which is actually pretty far north and has very cold winter temperatures for this species. But it is present in what is called Scottsport, just on the north coast of Balmullet. There is some modeling done by Chris Yessen in the UK. I think it was 2015. He had a couple of papers where he was talking about habitat suitability and where certain kelps could potentially live. And the westward islands off of Ireland's coastline, especially in the Mayo, Galway, and Kerry region, are predicted to be suitable habitats for Laminaria ochrolouca, the golden kelp. So that may not be the only record. We're not entirely certain how it got there, if it's been there for a very long time. But we have been repeatedly sampling tissue from the individuals that make up that very sparse forest in Scottsport to see if population structure is shifting over time at all. And then we did some population genetics with that, trying to relate it back to Portuguese population. And Stacey, what do we
So this is what I was talking about, the complication of using genetic data generated in one lab versus another lab, because there's some subjectivity in how you call your markers. And so we were able to do a different type of calibration, and there's comparable genetic diversity, but with one population, it's sort of hard to do the types of comparisons that we were showing on the slides earlier to the other populations that have been previously studied. But I was just going to add to what Kate was saying. I think this highlights 2 really important things: that there's this population of Laminaria ochrolouca way outside of its range. What do you call such a species? Because it's not invasive maybe, and it's not native because it's from nearby, but what do you call a species that shows up in a new place? And that terminology is really loaded, and I think we need to have better terminology and better definitions for what we call these species that are showing up in places, because it's not like it's being brought by a ship from North America or from Asia to Ireland, but how is it getting there? And the second point I think that's really important is these natural history observations. So natural history is inexpensive apart from somebody's time, but it's not sexy enough for funding. And it's really hard to get natural history funding, but without these baseline data, how did it show up there? And this isn't about Ireland, but I'm currently in Virginia on field work, and we found this little nudibranch accidentally, and it's the furthest record north. Is it a migrant that's following, a thermocline? But it's also really rare. So is it just that people haven't been doing monitoring? And so without just going out and enumerating taxa and knowing where stuff is and whether it's a healthy population or not a healthy population, those data seem par for the course, but without them, you can't do any of the fancy satellite or genomic technologies that we have access to are meaningless without those types of natural history observations. And so we really need funding agencies and other types of granting agencies to really prioritize those types of observations. They're cheap too. So
we need to get more boots on the ground or flippers in the water is what you're getting at.
yeah.
Exactly.
And
That's where the Joni Mitchell quote comes in.
Right.
Well, I think it's important also with taxonomic expertise to be able to identify seaweeds properly. And that's it's less of a problem in rocky shores, but we do a lot of work in mudflats, and there's not the same phycological expertise that there is in other places. And so a lot of invasive species have gone unnoticed because it's just a seaweed. And it's something that an invertebrate is eating or a fish is using, and it doesn't matter what it is. And so I'm sure that's the same for invert people as well. Like we need the taxonomic boots on the ground and flippers in the water.
Great. So last question before we wrap up, and I want to turn this over to Aaron. So I know sounds like Kate was able to get you out into the water a few times, which I'm sure was enjoyable. Nice warm water up there in Ireland, of course.
Well, I wouldn't go that far. Thankfully, I've kind of fully equipped of my own sort of, we'll say, blubber coating, so it was okay. It wasn't so bad for me.
So I'm curious if you have done some sort of like accuracy, ground truthing or water truthing to how accurate this bathymetry method that you've been using. Like, are you seeing like 70, 80% accuracy, 90%? Like what sort of accuracy is that?
No,
doing?
Yeah. So no, we haven't. We're talking to the National Parks and Wildlife Service, who've done a full sonar survey up at Kieran Bay, isn't it, Kate?
Kieran, yes.
So they actually have a really solid. They have a really solid data set of ground truth data that I can actually do a formal bake-off against. But what I can say is qualitatively, we gathered enough information to demonstrate to the funding agency that it was worth investing in and following up in. But for sure, as soon as we have something meaningful to show, it's going to go straight into publication, because nobody's I guess one of the interesting things maybe didn't really come across in the talk, that we are talking terra incognita here, and I guess it follows on with exactly what Stacy said as well. Nobody's ever looked at this thing before in this part of the world before. In every sense of the word, both for a genetic level, from a population perspective, and from a remote sensing perspective. So it is all it is new and novel. And it's also kind of essential because, and maybe I'm a little bit biased here, but to some extent, Ireland is like the canary down the mine in so many ways, in terms of this kind of, it's got its finger on the pulse of what's actually happening to the planet. And I just think it's a really interesting opportunity to use this as a sort of a test case across the board. Everything that you've heard today across the board as a sort of an exemplar about how we can sort of plan for the future and try to make get some sort of a sense of what's actually happening now and how we can try and save things. So, to get back to that Joni Mitchell quote.
Great. Well, thank you all so much for your time and for the valuable work that you're doing. Aaron, I'm very interested to see that paper when it comes out. I think that will have a lot to contribute in a bigger way. So please shoot that to me when you get it published.
Sure. Sure.
And Kate, Stacy, Aaron, thank you so much. I know everybody's on different projects and different times, and thank you all in the audience who stuck around for the remainder of this, and we will hope to see you on November 17th for our next webinar. So thank you all. Have a great day and evening.
Thank you.
Cheers.
Thanks for having us.
Yeah,
All
absolutely.
right.
Bye.
Bye.